I think one of the most interesting aspects of cell theory is not exactly a tenet of cell theory at all. It's the idea of spontaneous generation. It's an idea that existed long before cell theory was established, and the idea continued to persist throughout the centuries that cell theory was becoming, kind of grounded in scientific thought. The proposal of spontaneous generation was that life just kind of sprung into existence. It could come into existence spontaneously, often appearing from matter that was decaying or old, things that you would think of as, rotten. So that could be something like maggots coming from old meat that had been lying out on the counter, so flies were spontaneously generating. Or one of my favorite, I was doing some research on spontaneous generation, and I ran across old recipes for life, and one of the recipes for life said if you put old dirty clothes and wheat or grain in a barrel, you'll get rats. And so they had recipes for making rats or mice appear. So mice from sweaty clothes would be another example of spontaneous generation. I know this idea seems really funny to us now. We know that life does not spontaneously generate, but this was a hotly contested idea in the 1500s and 1600s when, early work was being done with microscopes and microbial life. Italian physician and poet Francesco Redi conducted an experiment to see if flies would spontaneously generate from particles in the air or from the flies themselves. So he left 3 chunks of meat out. One was exposed to air and flies. One was covered in cheesecloth, which is a fine mesh cloth, to expose the meat to air but not flies. And then finally there was a sealed jar to prevent both air and flies from passing into the jar to the meat. And sure enough, it was only the first chunk of meat that yielded flies, the one that was exposed to both air and flies. It was exposure to the flies, not the air, that was the deciding factor in whether or not life sprung into existence on this meat. So even though this experiment was extremely clear, spontaneous generation continued to be a point of debate among the early scientists. Well into the 1800s, this was a hotly contested issue, even for some of the scientists that laid the groundwork for what we now know as cell theory. So who were these people and what were these technologies that allowed us to lay that foundation, what we now know as cell theory, our, 3 concrete ideas about what cells are? Well, early on, microscopes were made mostly by people that worked with lenses. And so there was a father-son team that were spectacle makers, and they made some of the first lenses used in compound microscopes, or not even compound microscopes, just simple microscopes. And then Antony von Leeuwenhoek devised this little device, and so this was what he used to focus in on small drops of water, and in those drops of water he could see, he left peppercorns soaking in water and was able to see lots of little living microbes. I've seen some writings about there possibly being letters that he wrote to people saying he looked at his diarrhea, and there's lots and lots of things that he looked at under this microscope, but in all of them he saw little bacteria and little protozoans, and he called those animalcules for tiny animals. In the same time period, so we're talking about the 1600s, Robert Hooke was also looking at thin slices of cork, and he named what he saw cells. He was talking then about the monastic cells that cloistered nuns used, and, every nun had their own little cell, or what we would now think of as prison cells. But what he was really describing or seeing were the cell walls in this dead plant tissue. He had sliced the cork very thin, and so he was able to see the little rectangular boxes that were the cell walls around what used to be a living cell. So he gets credit for naming the cell. That name stuck, even though it took hundreds of years, over 100 years from that point, to develop microscopes that were able to see cells in more detail and with more confidence. So here we are at this point in the development of cell theory. We have two major scientists, one that saw the living animalcules in the droplet of water, and one that was looking at the cork tissue and named cells. In the 1800s, it was primarily German scientists leading the way in cell theory. They were sort of colleagues. They were in communication with each other, and they were Schleiden and Schwann. And they were looking at huge numbers of plant and animal samples under more modern microscopes of the 1800s. Schleiden was the one that proposed that all plants must be made of cells, and so he published his finding, and using that information and what Schwann was looking at, which were animal cells, he was prepared to publish what is now the first published tenet of cell theory, that all living things are made of cells. There must have been a lot of argument and debate between these two men. They were both from the same country. They both relied on the work of each other, because one was looking at animals and one was looking at plants. But only Schwann gets the credit for being the first publisher of any tenet of cell theory. And the way I've heard it explained, and my take on it, is that Schwann kind of ended up on the right side of history. He did not believe in this notion of spontaneous generation, and Schleiden did. And so Schwann was both the publisher of the statement, all living things are made of cells, but also he was on the correct side of the debate about spontaneous generation. So he had a little bit more insight, if you want to look at it that way. The next statement to be added to cell theory was that the cell is the most basic unit of life. That's talking about both structure and physiology. So the chemical processes going on inside the cells, that was the smallest unit you could get, the most basic, and still have the characteristics of living things. Eventually, spontaneous generation also retreated into our scientific past, and it was replaced with the notion that all cells come from other living cells. And this was actually published by a scientist named Virchow, and he published this notion, omnis cellula e cellula, that all life and all cells come from cells. So he proposed the final statement of cell theory. So that's the three fundamental tenets of cell theory. We know a lot more about the biochemical lives of cells now, so really we could add more statements to this list, but we have plenty of time to talk about that later on. The last thing I do want to talk to you about in this cell theory lesson is how the debate about spontaneous generation was finally laid to rest. We owe it all to a scientist whose name is one of the most recognized of our time, Louis Pasteur, the man that also developed pasteurization of milk and began the development of germ theory. It is not every scientist whose name is so fundamental to history that it becomes a word that is lowercase. It's not even a proper name anymore. Pasteurization. That actually is a great honor to reach that status. And so here, like so many experiments designed to show these like almost invisible forces of nature at work, Pasteur's experiment is a really beautiful one in the simplicity of the experiment. He used a swan neck flask, so they were able to do this. He used a Bunsen burner to heat the broth, but the swan neck was also created by heating the glass and pulling it out into this swan neck shape. So a lot of that was done by the scientists themselves. And so once that broth was heated and boiling, it was sterilized. And so he could let this flask sit for hours, days, weeks, months, years, and it would remain sterile as long as dust was being trapped in that swan neck flask. So air could still travel in and out, but any particulate matter, due to gravity, would fall down and it would not enter and be able to be exposed to the broth. But in his experiment, and there are various versions discussed, this is one of them, where the broth is tipped so that it interacts with the dust, and then you tip it back over, and either in a few hours or a few days, more likely, depending on the heat of the room and which bacteria you'd been exposed to, you get this sort of cloudy look to the broth, which you see right here. So that's one way of looking at it. You could also break the neck off the flask and expose it to the open air, and the same thing would happen. You would get this cloudy broth, and this could have been any kind of chicken stock or beef stock, but it was nutrient-rich. Microbes loved to grow in it, but life would not spontaneously generate. It would only form when life, other existing life was able to interact with that broth. So even the tiniest of organisms come from other living organisms. I know it's not a novel idea now, but in the 1800s it was big news. There was even a competition at the time being sponsored by the French Academy of Sciences, and this was the experiment that won that competition. And so it goes down in history as finally settling the debate about spontaneous generation. Louis Pasteur.